냄새 맡는 뇌, 비식. 비식은 이 뇌의 아래쪽입니다. 뇌의 아래쪽. 여기가 다리뇌죠. 그죠? 폰스, 다리뇌. 여기가 숨뇌. 뒤쪽에 보이는 소뇌. 여기가 측두엽. 측두엽. 여기가 측두엽. 여기가 측두엽이죠. 측두엽. 앞에는 전두엽. 이렇게 되고, 이 전두엽의 아래쪽에 여기에, 여기 코가 이렇게 있고, 코천장에 냄새 맡는 신경세포들, 후각수용세포라고 해놓은 거, 후각수용세포들이 요 코천장에 있고, 이 코천장에서부터 축삭을 내가지고, 코천장에서 축삭을 내가지고, 후각망울로 보냅니다. 요게 후각망울. 이게 후각망울로 보내고, 후각망울에서 이제 요 뇌의 아래쪽으로 이래 온다는 거예요. 아래쪽으로. 이 아래쪽이 여기입니다. 이 아래, 이렇게 아래쪽이죠. 이게 후각망울이고, 후각망울이 뇌의 아래쪽에 있고, 그 다음에 이렇게, 여기가 다, 냄새를 맡는 후각뇌입니다. 여기가 후각뇌. 여기 후각망울이고, 이렇게 와서 이게 다 후각뇌. 그러니까 결국 이렇게 해당되는 이 부위라는 겁니다. 이 부위. 냄새, 그러니까 1차 후각피질은 여기에 있습니다. 여기.
The olfactory system is responsible for the sense of smell or olfaction. Basically, airborne molecules emitted by an odorant source are detected by olfactory sensory neurons located at the roof of the nasal cavity. These neurons convert chemical stimuli into electrical signals and send them via the olfactory nerve to the olfactory bulb, then to the brain, where they are interpreted as odors. Odorant molecules are first dissolved in the mucus secreted by the olfactory epithelium, which guides them to the cilia of olfactory neurons. This is where odorant molecules bind to their receptors. Each neuron expresses a single type of protein receptor. There are only about 400 different receptors in humans, but they are used in a combinatorial way such that one odorant can bind several receptors, and one receptor can bind several odorants. This enables the olfactory system to recognize an enormous number of odorants. Odorant receptors are G-protein coupled. Upon binding to the odorant, a signaling cascade is activated, leading to membrane depolarization. When the olfactory stimulus is strong enough, action potentials are generated and conducted along the axon to the olfactory bulb. The axons of all olfactory sensory neurons form the olfactory nerve, also known as cranial nerve 1. In the olfactory bulb, these axons synapse with second-order neurons, the mitral and tufted cells, within structures called glomeruli. Each glomerulus receives axons from sensory neurons that express the same protein receptor. The second-order neurons are stimulated by sensory neurons, but they also receive inhibitory feedback from the cerebral cortex. This means an odor can be perceived differently under different circumstances. For example, the smell of food is more appealing when one is hungry and is less so when one is full. The axons of mitral and tufted cells form the olfactory tracts, which project directly to the primary olfactory cortex. The primary olfactory cortex is not one, but several cortical areas located on the base of the frontal lobe and inferior surface of the temporal lobe. These primary regions then project further to some other areas of the brain, mediating different aspects of odor recognition and response. Because olfactory neurons are exposed directly to the noxious external environment, they are replaced more often than other neurons. Stem cells in the epithelium differentiate into new olfactory neurons, whose axons grow along the existing axons to the olfactory bulb. Any factors that destroy all olfactory neurons at once would result in a permanent loss of sense of smell, a condition known as anosmia. Illnesses that cause inflammation of the nasal mucosa may lead to transient anosmia. Loss of smell also affects the taste experience, as taste and smell are the two aspects of flavor. The ability to smell decreases with normal aging, but anosmia is also an early sign of several neurodegenerative disorders. Because epileptic seizures often originate from the brain area associated with the olfactory cortex, seizures are often preceded by hallucinations of disagreeable odors.